From the American Neurological Association, I'm Adeline Goss. This is ANA Investigates. There are trillions of bacteria living inside each of our gastrointestinal tracts, and there's growing evidence that those bacteria communicate with the brain. Some researchers are even hopeful that drugs targeted at the gut microbiome could help treat diseases as diverse as Parkinson's disease, multiple sclerosis, or even autism. The nature of this so-called gut-brain axis, the extent to which bacteria might actually shape brain function and disease, is the topic of our show today. Dr. Sarkis Mazmanian is the Louise and Nelly Sooks Professor of Microbiology in the Division of Biology and Biological Engineering at Caltech. He's the recipient of many awards, including a MacArthur Genius Grant, for his work on the gut microbiome. Dr. Mazmanian has co-founded two companies, one working on the gut-brain connection. You can read more about that in our episode disclosures. Welcome, Sarkis. So nice to speak to you here on ANA Investigates.
Hello, Adeline. I'm very happy to be here.
So, Sarkis, we'll talk about some of the details of your research and of this field in a moment. But first, I wanted to ask a big picture question. What do you see as the potential therapeutic impact of understanding and eventually manipulating the gut-brain axis?
Adeline, there's likely many potential answers to that question. I'll highlight the word potential because we obviously have not achieved any therapeutics based on the microbiome for any indication that's related to neuropsychiatric, neurodegenerative, or neurodevelopmental disorders. But what I would say, again, from a big picture standpoint, is that modern pharmaceutical approaches oftentimes require that the drug crosses the blood-brain barrier to enter into the brain to have its effects, which is very challenging to get drugs into the brain across the blood-brain barrier because there's a reason why our brain is protected, obviously, and therefore this creates a challenge in terms of both dosing and side effects. And what I would contend is that the microbiome over many millions of years has figured out how to deliver molecules into the brain. In fact, we know that this is the case. And if we can harness the knowledge that microbes have built and use that for drug discovery and drug development, I think that this would be a game changer, in my opinion, in how we treat various different disorders of the brain.
Great. Thank you, Sarkis. So there are a number of neurologic diseases where we see that patients' gut microbiomes look different from healthy people's. What is the spectrum of neurologic diseases where we see those kinds of associations?
To date, there have been associations between microbiome changes and various different conditions that include depression, Parkinson's disease, autism, schizophrenia, and Alzheimer's, and a host of others where there's, I would say, some evidence, but the data that have not been widely reproduced in a number of different cohorts. And even among the conditions that I just mentioned, it's clear that, at least to me, Parkinson's and depression appear to be neurological conditions where there's reproducibly a very robust effect on the microbiome. And so, again, this is my opinion, and it may be different people may view this question differently. But again, I don't think it's all neurological conditions where the associations are robust, but a handful, namely Parkinson's and depression.
So then a second question, we see these associations between diseases and the gut microbiome. In which conditions do we see the strongest evidence that there's an actual causal effect where the gut microbiome seems to be shaping central nervous system activity? And what is that evidence?
In humans, I would argue, Adeline, that there is no evidence to show a causal effect. And by causal, I mean that the microbiome is directly contributing to symptoms, pathology, or any other aspect of a diagnosis of a neurological condition. In 2023, we're still at the point where we have a lot of correlative evidence, mostly through microbiome profiling of stool, that there are changes in the microbiome. And some of those changes appear to make sense in terms of the fact that there are organisms which may be contributing to neurological conditions in ways that have been worked out. But in humans, the actual evidence to show a causal relationship, which would require some sort of intervention, either removing or adding microbes or dietary changes or some other way to directly alter the microbiome and then see effects on the disease, that data is still not convincing, though there are many studies underway to address this really, really important question. Adeline, in animal models, I would argue that there's robust evidence, that there's overwhelming evidence that the microbiome actively and directly contributes to both symptoms and pathology associated with many, many neurological conditions. In many animal models that, again, reproduce aspects of human neurological conditions, what investigators, our lab and many other labs around the world have done are taking the approaches of completely removing the microbiome of animals, and by animals, most of that research is in mouse models, where we can either treat the animals with broad spectrum antibiotics and then behavior test the animals, or rederive the animals under germ-free conditions, which means that the animals are placed into sterile bubbles, into these chambers where there's no bacteria, viruses, or fungi, so they're microbiologically sterile. And so then in the absence of a complete microbiome, people have shown, again, in many, many animal models that those disease endpoints change. People have added specific probiotics to animal models and shown that by just adding one particular organism, or in some cases a handful of organisms, that those disease endpoints in mice can change. And there have been studies where fecal transplants have been performed in mice where, let's say an animal model of a neurological condition where the microbiome is altered, a healthy microbiome or a quote-unquote normal microbiome is transplanted into those mice. And in some instances, it's been shown that essentially repairing the microbiome leads to improvements in symptoms associated with neurological conditions.
And just to make this a little more tangible for those of us who aren't in the field, what is a specific example, of a mouse neurologic disease that seems to be shaped by the mouse gut microbiome?
I'll refer to work done in Parkinson's models where, again, the effects of the microbiome appear to be quite robust, where in one particular animal model, there's a transgene, meaning an overexpression of a human protein called alpha-synuclein. Changed alpha-synuclein are widely believed to be causal in Parkinson's disease. And so in a mouse model, people have overexpressed human alpha-synuclein, and that animal shows motor symptoms, which is a hallmark of Parkinson's disease, but also inflammation in the brain and then constipation-like phenotypes in the intestines, where antibiotic treatment or germ-free regeneration results in those symptoms going away. So mice that have the transgene that express human alpha-synuclein, if they don't have a microbiome, they don't develop motor symptoms, they don't have constipation-like phenotypes, they don't have neuroinflammation. And more particularly, when specific organisms that are pathogenic, microbes that promote Parkinson's disease, are transplanted into these mice, so the mice are colonized with particular strains of bacteria which are elevated in human Parkinson's disease, those bacteria are able to reproduce the symptoms in the mouse model just by themselves.
Great, thank you. So walk me through how we think this works. What do you think are the major mechanisms by which the gut microbiome may influence the central nervous system?
So the brain and the gut are always in communication, right? Constantly throughout our day, throughout our lives. There are a number of different ways or conduits by which the gut and the brain communicate. Oftentimes, there are molecules that are exchanged between the two organs, so small molecules that circulate through the blood, either produced in the gut that wind up in the brain or in the brain that influence gut physiology, so that's one aspect. The other would be the immune system. So it's well known that immune cells traffic between the gut and the brain, so they're constantly moving between the two tissues and carrying information with them. There's a lot of evidence to suggest that immune cells can carry information from the gut to the brain, and when those immune cells are in the gut, are being primed or educated by the microbiome, and then that signal is transferred to the brain, not by the microbe itself, but by the immune cells that's been altered by association with the microbiome. And then in addition to immunity and small molecules, there are neuronal connections that link the two tissues. So there's the vagus nerve and spinal cord neurons that innervate the gut, meaning that they're found in the intestines, the neurons that are found in the intestines are connected by additional neurons to the brain. And so signals can be sent back and forth through the nervous system as well. And again, lots of evidence that the microbiome can essentially hijack this pathway to send its signals to the brain.
And is it thought at this point that there might be a single, like, bad actor bacterium in certain diseases, or more that there might be a constellation of bacterial activity that is together influencing the central nervous system?
Adeline, I think it's highly unlikely that any one organism is going to have a similar effect in all people, meaning whether it's a beneficial organism that provides resilience and protection from disease or a harmful organism that may promote disease. Oftentimes, those interactions between microbiome are personalized to the individual. And so it's my opinion, we don't really have the answer to the question, Adeline, but it's my opinion, it's going to be almost like a lock and a key relationship between microbes and particular people, specifically their genetic makeup. And gene environment interactions, or more specifically, gene microbiome interactions, are going to be prevalent in understanding how the microbiome contributes to a particular disorder. And so I'll go one step further than just the microbes themselves, because what's more important isn't the genus or the species of the organism, but what function does that organism have? What genes does the microbe harbor, and how are its metabolic and functional capabilities interacting with the host? I think this is where the research is going, is to try and understand how the function of a particular microbe influences its relationship to a particular disease, rather than just what the name of the organism is.
So are we at the point yet where we have a sense of what a quote-unquote healthy gut microbiome might look like vis-a-vis the nervous system? For example, I have heard that there were ideas that taking a daily probiotic or changing your diet might prevent or treat different neurologic diseases. Are we at that point yet, or would that be getting too far ahead of ourselves?
I believe we're at the point where we can begin to define a healthy microbiome. I don't think we're at the point where we know whether there are interventions like diets or probiotics that are going to prevent or reduce neurological symptoms. So the second half of your question is that's the cutting edge of the research, and that, those answers are still forthcoming in terms of what we can do in humans. However, there's a lot of evidence that helps us correlate health to the microbiome. And this comes from the many, many different microbiome profiling studies that are already in the literature that are comparing, let's say, disease X, whatever it may be, to a healthy control population. Oftentimes, these studies show that individuals who don't have a diagnosed disease have more diverse microbiomes. And what I mean by that is that they just have more species of bacteria and viruses and fungi compared to a companion disease population. And what we also know from just profiling human microbiomes outside of disease is that diverse microbiomes are found in people who eat plant-based diets, and very diverse plant-based diets. So people who eat many, many different types of plants or plant-based foods results in more diverse microbiomes. So if you can sort of connect those dots, that a diverse microbiome correlates with health, and a diverse diet contributes to a diverse microbiome, it helps us understand that diversity in terms of the microbiome and in terms of what we eat are associated with healthy outcomes.
Great. So we've been talking about diet. I wonder what other potential mechanisms have been considered to try to treat neurologic disease through the gut microbiome. Are we talking about fecal transplants, antibiotics, probiotics? What specific treatments have been considered?
I think all that you mentioned have been considered, but there are pros and cons to each. I don't believe long-term antibiotic use is going to be effective in treating any condition because of the negative consequences that come along with depleting your microbiome. People are trying fecal transplants for a variety of different conditions, including neurological conditions. The jury's still out on how effective those are. And of course, there are probiotic and prebiotic treatments that are being tested. Probiotics are the organisms, prebiotics are the dietary fibers that help particular organisms colonize and thrive. And those have been tested as well. you will be able to find companies and other entities that make rather strong claims about the ability of their products to improve neurological conditions. Those claims have not met the rigor required for, let's say, FDA approval or for societal approval, in my opinion, just yet.
And how close are we, do you think, to developing actual effective treatments for neurologic disease through some of these mechanisms?
I'm quite bullish, Adeline, about our being the biomedical community's ability to have an FDA-approved drug, which again is the gold standard, in the next 5 to 10 years. There's just so much interest in this space and so much potential for the microbiome to affect our biology. But again, I want to also emphasize that it's important to not rush this process to make sure that we're being responsible in the drug development campaign, just so for the very obvious reasons that we don't cause any harm to people or the side effects are worse than their symptoms, but also so that the microbiome field has the credibility it needs to continue.
Yeah, so really important to be responsible so that we can get it right. So Sarkis, just a technical question. We have, tens or hundreds of trillions of bacteria in our GI tracts and hundreds of different bacterial species. What breakthroughs in recent years have allowed us to study a system this complex?
The major breakthrough, which I believe enabled the microbiome research, is DNA sequencing. And a lot of the DNA sequencing approaches that have been applied to microbiome research have come on the heels of the Human Genome Project. And so that's both in terms of the actual sequencing itself, but also the bioinformatics required to understand the sequencing data, essentially to put the pieces together, whether it's assembling a genome or putting together pathways of genes to impute function. This really has been the catalyst for springboarding the microbiome field. More recently, additional approaches such as mass spec technology, mass spectrometry, which allows us to detect small molecules and even large molecules, which are the products of the microbiome, so that we can we can understand what is the molecular exchange between microbes and our cells. That technology has really accelerated research because, again, it's gone beyond just the description that microbiome sequencing, DNA sequencing can give us, to actual molecules which are performing the functions. And so that's been a huge advance. And also in improved animal models. So we now have the ability to manipulate the microbiome in mice and other animal models, which give us insights into biology and to function that, again, give us clues in the mice that are testable in humans. So again, I'm quite enthusiastic about the next several years of microbiome research and hopeful that it's going to pay off in the clinic.
Thanks, Sarkis. So it sounds like there's a lot of excitement on the horizon, and I wonder if you could just speak to some of the major questions that remain to be answered in this field, specifically if you think about a listener to this episode who might be a junior researcher interested in the gut microbiome.
And I think it comes back to the causality question, right? Because it's become easy to measure changes or differences between individuals or populations, again, stratified in many ways that say disease versus healthy individuals. But unless we know whether or not the microbiome and particular organisms or molecules in the microbiome are contributing or even causal to those outcomes, then we're not in a strong position to improve human health. I think a lot of research, in my personal opinion, should be focused on is to move away from correlative research and to emphasize our efforts in actual interventional research so that we can get to this cause and effect question.
Well, thank you, Sarkis. It was really enjoyable to talk with you about this, and I think exciting to consider another potential avenue for treating really tough to treat central nervous system diseases. Thank you so much.
Thank you, Adeline.
That was Dr. Sarkis Mazmanian, professor of microbiology in the Division of Biology and Biological Engineering at Caltech. ANA Investigates is the official podcast of the American Neurological Association. The show's producers are members of the ANA Education Innovation Committee, chaired by Megan Ritchie. I'm Adeline Goss, podcast host and executive producer. Our program was produced today by me, Caitlin White, and Sarah Stern-Nezer, with help from Jen Hurley. You can listen to past episodes of the show, subscribe to the show so you never miss an episode, and access all of the ANA's educational programs on the Education tab at myana.org. That's it for ANA Investigates. See you next month.